Hey everyone, okay, we're going to try to go super fast through this because I covered a lot of this with our nucleic acid talk, but I want to kind of bring our focus back to nucleic acids. We're talking about DNA. Oh, what are we talking about? Protein synthesis, how proteins are made. So why are we talking about DNA? We'll find out. Okay, so just bringing your mind back. Here's DNA. It's like a double helix, twisty ladder. How do we get to that point? Well, here's our cell, which we just talked about. Inside the nucleus of the cell, we can find our DNA chromatin or chromosomes, and that's really wound up. And if we zoom in even closer and unwind it to the max, then we have our DNA. DNA is packaged up in chromosomes, or it's hanging out as chromatin. We talked about the parts of the DNA molecule. It's called a nucleotide, and they have a sugar phosphate base. And those four bases, and this is where it's going to be important, are called adenine and thymine, A and T, and C and G, cytosine and guanine. And you can think of it as apples in the tree and car in the garage, or pointy letters and round letters go together. It is going to come and be important. It will. Here's another picture of our nucleotide. Here's our sugar at the home plate, phosphate, our baseball, and our base. It doesn't look like a mitt in this picture, but the mitt, like the guy getting excited, we won. The order of the bases is what makes you. So these bases here. This backbone, the sugar and phosphate, is the same. If it's DNA, it's deoxyribose and phosphate. That's the backbone. But the inside of the ladder, this part, the colorful part, those are made of these bases, A, T, C, G. The order that they're in is the important part. That's what's important for protein synthesis. Okay. DNA copies itself, by the way. It can unzip, and then the bases get matched up by an enzyme, by a protein, by the way, by an enzyme. And then it's just copied. That's how DNA gets copied before mitosis happens in interphase. See how these things are connected to each other? That's why I love biology. So that little enzyme, boop, it'll just pop in and it will just zip down, literally unzip down and copy and match up the base pairs. So how does that happen? It unzips, we match up the bases. So if we wanted to go through, what would C match up with? Oh, don't you love the sounds? Here's our enzyme. It's coming by. It's a matching enzyme. It's a building enzyme. DNA polymerase 3. It's going to match. It's going to see this letter C base, cytosine, and it's going to say, round, C, car, garage. I need a G. And then it's going to work its way down the DNA molecule and match them. Why is the other one upside down? Is that a mistake? Nope. One side kind of would run what we would consider right side up, and the other's upside down. It's a molecule. So for us, we just kind of draw it upside down. T, let's see, that little enzyme is going to see the T, and it's going to say, what's pointy? Tree, apples. It's going to match an A. G is going to match with C. A is going to match with T. And this final G, I don't have it up there, but that would be another C. So this is where we're going to get into protein synthesis. I just wanted to point that out, review with you what a nucleotide looks like. That A and T, C and G match up, those are the bases. Now, here's where we're going to get into protein synthesis. This can be incredibly complicated. We are going to make it simple. Okay, we really are. We have DNA. DNA can copy itself through a process we call replication. We have RNA, another nucleic acid. RNA is produced from DNA, messenger RNA, through a process called transcription. And then this RNA will go to a ribosome and produce a protein. That's called translation. And then those proteins, they make your traits. So that's sort of what we're going on. We're going from nucleic acid language to amino acid language. That's what's happening. We're translating from the language our blueprint is in to the language of proteins. I'm going to link a video to this because there's some really excellent videos that show this. So what's this transcription stuff? DNA is this huge molecule. It's like your book. If I told you go study your book, Well, this is huge. I mean, like, there's over 600 pages in this book, almost 700. Where do I start? What do I do? But if I said, go into your book, go into chapter 3, I had to remember, it's chapter 3, go into chapter 3 and read about protein synthesis. Well, now I'm very specifically telling you where to find this information. It was just here, I just saw it. It's at the end. Specialized cells? Yeah, it's after. But anyway, sorry, I know now I'm like distracted. But if I went there and I was like, hey, look at this, you'd find this information on page 54. Well, now I'm more specific. I'm into a smaller section. I know where to study. But maybe I wanted to send you home. Maybe there's people that don't have a book, or maybe I'm like, we're only going to use the book a tiny bit. I'll just make copies, which is not allowed to do that, and I do not do that. Okay, but let's just say I did. I didn't. I'm not. I promise. I'm not just saying that for the video. I really have not done that. But let's say I did that. Well, I could make a copy of these pages and send them to you. And then you would take notes on them and write them in whatever you're going to write. So you've transcribed. Literally, that's what that word means, notes. So what's the point? So we have our whole DNA. It's like the giant book. Well, I don't need every chapter, everything, every page. I need maybe just 1 or 2 pages from the book. In transcription, I don't need the whole DNA. I need one recipe, one protein. So the DNA is going to get opened up, and that one page, just like how I told you, like page 54, is going to get copied. That one section of DNA is going to get copied. The copy we make, well, just like the copy, if I made a copy of this page, maybe instead of 54, I'll make 56 because there's a nice picture on there. Maybe the copy I make of this is on different paper. Maybe it's in black and white instead of color. They can get crumpled up and messed up. I still have the original. That's why a copy is made. Your DNA is so important. It's not, and so big, it's not going to leave the nucleus. But I can make a little copy and it can leave the nucleus. That's transcription. A little messenger RNA copy is made from DNA. Okay, that's what this picture shows. What's this mRNA thing like? Well, here in red we have our DNA, double helix, double-stranded, ATCG. We've got our sugar phosphate backbone. The sugar is deoxyribose. What about RNA? Sorry, I just realized I said that really fast, but it's all there. RNA is single-stranded. The backbone is also a sugar phosphate backbone, but the sugar is ribose. Inside, it's not matched up with anything. We have C and G and A, but instead of T, we have uracil. Remember, that's like our pinch hitter. That's our like substitute. Our quarterback is out because he was just crushing the other team, and so they put in the backup quarterback. That's what uracil is. It's the backup for T, for thymine. Okay, and it's generally much smaller than DNA. It's just a copy of one section. It's not a copy of the whole DNA. It's a copy of one recipe. Let's just call it the recipe to make melanin, since that's something that a lot of us are familiar with. So DNA gets unzipped. An enzyme comes along and matches the bases together. T will match with A will match with U in this case, C will match with G, and G will match with C. So that's what happens. So we're not going to go through all that. Now, here's how things work. All right, let's kind of take a look. We have a lot of living things, and the nutrients are the same. We've talked about those. These are amino acids, but they're carbs, lipids, proteins, nucleic acids. All living things are made of cells. Here's a cell. It's moving. It's alive. And inside all the cells, we have DNA blueprints. No matter what living thing we're talking about, it has those blueprints. And then those blueprints make you. Look how cute you are. Okay. We can get tons of different living things. A plant, a bacteria, a you, whatever, a kitten, with the same nutrients and the same molecule, DNA. So how does that happen? Think of it like this. We have raw materials, like wood and nails and stuff. We get a blueprint. We have tools. We can make a product, like a doghouse. Well, for us, those raw materials are not wood and nails. They're carbs, lipids, proteins. Our blueprint is the DNA. Our tools are things like enzymes. We have our product. Instead of the doghouse, it's the dog. He's in the doghouse. I guess that's why he's sad. So how do we get so many different, just people, not just animals and stuff? How do we have so many different people? Well, I can take the same materials. If I have two different blueprints, I probably need the same tools. I can make two products, like a doghouse or a table and chairs. It's the same thing with living things. We take the same basic building blocks, but we have two different blueprints. We have the same or very similar tools, and we make two different things. So how do we make those things? How do we make those proteins? Well, that's protein synthesis, sometimes called translation. That's the process of this messenger RNA leaving the nucleus. Goodbye, fam. I'm grown up. I'm going away to college. The ribosome. I've left the nucleus and being turned into a protein. So how does this happen? Okay, well, let's take a look. So this RNA thing, this messenger RNA, was made through this process called transcription. DNA opens up. The bases get matched. Bam. That's all you have to do. That's how we make our messenger RNA. The messenger RNA then leaves the nucleus, it goes out a little pore, and finds a ribosome. A ribosome is a protein factory. So how does it work once it gets to this protein factory? Well, let's kind of get some things clear. This messenger RNA, that looks a lot like DNA. The ribosome, that's the blue thing in this picture, is made of protein and something called rRNA. That's just the bluey turquoise background thing. That's the ribosome. That's the protein factory. So now how are we making a protein? The protein factory, ribosome, reads the messenger RNA like we would read words. It reads 3 letters at a time. And there's a word for that. It's called a codon. So it reads these 3 letters at a time. AGU, CGU, ACG. It's reading those. Then, as it reads them, the messenger RNA moves over and something is attracted to the ribosome. It's this weird shaped molecule, excuse me, that's called tRNA, transfer RNA. Well, what's on there? On this little golden molecule? Well, one, look, we have matching letters. A and U match, C and G match, G and C match. So it's got the match. And it comes along with a colorful bead. This colorful bead is not a bead at all. It's one of those 20 amino acids from our protein talk. So it takes one of these amino acids, and then they get joined together. As this thing moves along through the ribosome, the ribosome's job is to grab the correct transfer RNA. Where are those? They're just sitting around? Yes, they're floating around in the cytoplasm. The ribosome grabs the correct tRNA. It has the correct amino acid attached. And then the ribosome's other job after it grabs the tRNA is to connect the amino acids together with a peptide bond. That will form a polypeptide chain, which will then fold into a protein. That's how proteins are made. So I think I have this written over here. This little bit that matches with the codon we call anticodon. That's found on the transfer RNA. It's the matching codon. And then it just grows and connects them and makes a protein. That's it. So what was amazing was somebody figured out this code, this Rosetta Stone of DNA language. They figured out what letters match to what amino acids. So who cares? Why is that important? Oh my gosh. This is like breakthrough. This is why we have all the biotech that we have today, and why in the future, when you're older, you're going to have all these amazing different things that are going on thanks to biotechnology. Because we know what code matches with what amino acid, now we can look at DNA. We can look at DNA's code and say, here is a gene. This gene codes for melanin. This is where it starts. This is where it stops. And we can find the locations of those on the DNA. So we can understand now someone has albinism, meaning they don't make any skin pigment. Well, right here in the DNA, the code for melanin, it has a mistake, a mutation here that says stop, and it's not being produced. Here we have someone who has cystic fibrosis. That was one of the stories. Is that one of the stories here, or is it farther along? I don't remember. I remember reading the story about it, like super tragic story, by the way. Cystic fibrosis, super tragic. Here, right in this CFTR gene to make mucus, these 3 bases have been missing, and it's changed this whole code. So this person makes extremely thick, dry mucus. So now we understand what these codes in our DNA mean. Before it was just nothing. When I was in school, people said part of the DNA was junk DNA, like that it didn't do anything, which is crazy. It does. And now we understand what the different parts of our DNA do. So now that we understand what codes for what, now the area of technology is how can we alter the DNA code in living cells to get rid of genetic disorders? And that's all because we understand what codes for what. Incredible. That's why I like that stuff. So I hope this, I know it was kind of fast. I hope this helps you understand a little bit about protein synthesis. I will link in the description some other videos to protein synthesis about it happening. To summarize, DNA is our big book that we don't want to leave the nucleus. We don't want to use every code all the time. If this was a cookbook, I don't want to make every recipe for dinner. I don't have that much food in the house. But I don't want to make every recipe. Maybe I only want to make 2 recipes out of this cookbook. Maybe I only want to make 1 right now out of this cookbook. I go and find the correct recipe. Okay, so DNA gets opened up just like I just opened this book. We find the recipe we want. That's the gene. So we find the correct code we want, the correct gene. Well, let's pretend it's right here. Here it is. Well, I don't want to take my cookbook, my special cookbook, out into the kitchen. I'll just jot down the recipe onto like a Post-it or something. Okay, I've got my recipe. Now I can close up my book. DNA gets closed back up. Now I can take my written recipe on my Post-it, my messenger RNA, and I can go to the kitchen. That's the ribosome. So I'm going to take my recipe, go to the kitchen. I can look on it if this gets all egg and whatever on it. I don't care. I'll just throw it away. I've still got my recipe in my book. I've still got the original gene in the DNA. If messenger RNA ends up getting messed up, whatever, I can always make more. I can always check the recipe again. So I've got my messenger. I'm at the kitchen. That's the ribosome. So now I get my ingredients. Those are the amino acids. And I bring them in the correct order according to my recipe, according to my messenger RNA. Okay, so I'm going to bring in eggs, and I'm going to get vanilla and sugar. I've got my wet ingredients for whatever I'm making cookies, I guess. And I mix them in the bowl according to the instructions on the recipe. So how is a protein made? This recipe, this messenger RNA, comes to the ribosome, the kitchen, and the ingredients are added in the correct order that is said on the messenger RNA. The amino acids are brought to the ribosome in the correct order that they are in the messenger RNA. Then the ribosome's job is to add them correctly in the correct order and connect them together. Then we have a protein. That's all you need to understand about protein synthesis. Like I said, I'll add some videos, but you don't need to understand the process in detail. DNA, messenger RNA. Messenger RNA to the ribosome. Ribosome grabs the tRNA that has the amino acid and builds it all together to make a protein. That's it. I hope that helped. I hope it didn't make it more confusing. But that's something that I'd be happy to talk about in class on Monday if you feel like we need more time on that. And I will talk to you later. Bye.